All right, so picking up about where we left off on last class, we just started talking about proteins, so I want to just start from the beginning of proteins. So proteins are the most abundant molecules in your cells. They make up a huge part of just the general biomass, is just how much stuff is in your cells. Well, 50% of the stuff that's in your cells is made of protein. They have a lot of important functions, a lot of which we'll go into after the test when we start talking about cells and cell components. They include structural support, so cytoskeleton, storage, transport, so that's also cytoskeletal elements, and control of transport, communication, so there's a lot of cellular junctions that are mediated by proteins. There's the exoskeleton, there's the kind of extracellular matrix, there's movement, so motor proteins interact with other kinds of structural proteins to cause movement and defense, as well as some other features as well that we'll go into over the next few chapters. Proteins are made of units called amino acids. So amino acid is the monomer that makes up the polymer of a protein. And amino acids are joined together to make something called a polypeptide. So a string of amino acids is called a polypeptide. Proteins can consist of just one polypeptide, so a single string of amino acids could be considered a functional protein, but a lot of proteins are made up of multiple strings of amino acids that are linked together, and we're going to look at that in a few slides in terms of what that structure is called. So here's what protein's monomer looks like. So the monomer of a protein is an amino acid. Amino acids get their name from their functional groups. So we talked about last class some functional groups. Amino group is a basic functional group. Carboxyl group is an acidic functional group. So this is called an amino acid. So it's got an amino group and an acidic group, carboxyl group. They both are on either side of a central carbon. On the top of that central carbon is a functional group called a side chain. And a side chain gives each amino acid a different function. So it is a functional group for an amino acid. So just to slide this up so you can see it a little better. So here's your side chain. Here are 3 example amino acids of the 20 amino acids. Glycine has just a hydrogen there. It's very, very simple. Cysteine here, a little bit more complex. Tryptophan, fairly complex, has a bunch of carbon rings. It has nitrogen in here. It has a bunch of mostly hydrocarbons, but arranged in 2 rings here. So dependent on what makes up the side chain, this is going to give the amino acid different properties. And then when that amino acid is part of a protein, it's going to give the protein different properties. And we're going to talk a little bit about that today. So you can have non-polar side chains, you can have polar side chains, you can have acidic side chain. So if you had a COOH up as part of the side chain, it would be an acidic amino acid. You could have a basic amino acid as well. So you can have lots of different properties to your amino acids. Okay. So just like in carbohydrates, amino acids are polymers, and they're polymers that are produced through dehydration synthesis. So here is down here 2 amino acids. This just gives it a generic amino acid because it just has an R here instead of actually drawing out the side chain. The carboxyl group of this amino acid has an OH. The amino group of this amino acid has an H. Dehydration synthesis is going to remove H2O, and you get formed between that carbon and nitrogen a peptide bond. So the bond between 2 amino acids is called a peptide bond, and this is why long chains of amino acids are called polypeptides, because they have many peptide bonds. Just like in carbohydrates, if I wanted to break down this, which is called a dipeptide right now, into an amino acid, I would do hydrolysis. So dehydration builds, hydrolysis breaks down, just like we've seen in carbohydrates. Yep. two amino acids. Bond between two amino acids in a protein is called a peptide bond. So you'll hear them referred to as polypeptides. So you've got dipeptide, polypeptide. Okay, so if I continue to link more and more amino acids together, again, just like linking monosaccharides together gets you a polysaccharide, linking amino acids together is going to get you a polypeptide. And that polypeptide is going to do different things. So let's just imagine I put, all right, here's amino acid, get a different color here, link to an amino acid here, link to an amino acid. Say I'm making this long chain of amino acids, this long polypeptide. And let's say, as part of this polypeptide, all these black amino acids are nonpolar, but the two blue amino acids are polar. So what does polarity mean? What does polar mean for a molecule? Okay, they're going to attract because polar means it has a charge, right? So remember, polar means it has positive and a negative partial charge. And two things with partial charges form what bond? What's the bond between partial charges? Hydrogen bond, right? So this would be hydrogen bond. Okay, so a hydrogen bond will form between polar side chains on polypeptides. This causes your proteins to fold. So here would be just a straight protein, but because you have two polar side chains, it's going to cause that protein to begin to fold. So we start to refer to proteins by their structure and levels of organization of structure. The primary structure of a protein is simply the order of amino acids. So just what order did we put the amino acids in? Did I put tryptophan before cysteine? Did I put serine before tryptophan? Like how did I arrange them? What order did I put those amino acids in is called the primary structure. The secondary structure, and I'm going to show you pictures of these on the next slide. Secondary structure is how that polypeptide folds due to hydrogen bonds between amino acids. So just like I was showing you here, hydrogen bonds form between polar amino acids, causing that polypeptide to fold. And you're going to see that there's two main ways that proteins fold. They either fold into a sheet, which this is starting to do. So if this kept folding like this, this would be considered a beta sheet. Say I had another polar amino acid here. So that's a beta sheet. It can also fold into an alpha helix, and I'll show you a picture of that, but basically that's just a spiral form. So rather than beta sheet, which is like this, with hydrogen bonds, a alpha helix is a spiral. So you have a spiral versus a sheet. Okay, so this will be the alpha. This would be beta. Okay, so secondary structure is how it folds. Then the overall shape of the polypeptide, and I'll show you a picture of this to help you with understanding of that. So basically how the alpha helices and the beta pleated sheets are arranged relative to each other in the overall polypeptide, that's called the tertiary structure. So you might have an alpha helix followed by another alpha helix followed by a beta pleated sheet, or you might have an alpha helix followed by a beta pleated sheet followed by an alpha helix. So what order those different secondary structures come in, That's the tertiary structure. Some proteins stop there. Some proteins are only single polypeptide, and that polypeptide folds, and that's it. That's the whole structure of the protein. Some that have more than one polypeptide, they're also going to have something called the quaternary structure, and this is the shape of the protein when two or more polypeptides interact. So you could have multiple polypeptides sitting on top of each other. That would make your tertiary or your quaternary structure. So primary structure, order of amino acids. Secondary structure, how those fold. Do they fold into a helix? Do they fold into a sheet? Tertiary structure, how are those helixes and sheets arranged relative to each other? So what overall shape does the polypeptide have? And then quaternary structure is how multiple polypeptides interact. Because some proteins are what are called complexes. They only function when you have multiple large polypeptides linked together. So here's some pictures to help with this. So primary structure, simply just the order of amino acids. When those amino acids are put together, dependent on the polarity of the different side chains, how it folds will cause the secondary structure, so alpha helix, beta pleated sheet. How those different secondary structures are arranged relative to each other causes the tertiary structure. So here you've got an alpha helix over here, you've got a beta pleated sheet here, you've got another beta pleated sheet here. You might have some other different kind of forms that are partway between. Here's another alpha helix. So that's the tertiary structure. And then this whole polypeptide is just one component of this final large protein. It's got this darker shaded purple polypeptide as well. So how the darker shaded purple and the lighter shaded purple polypeptide come together, that would be your quaternary structure. Okay. What do I really want you to get out of this? If you really understand the first two orders, that's probably enough for now. We won't really spend a lot of time dwelling on tertiary or quaternary structure, but knowing that the primary structure, the order of amino acids, leads to different folding patterns, that's really the main take-home. So the primary structure is just the straight-up order of amino acids. What do you think would happen to the secondary structure if I changed the order of amino acids, the primary structure? Yeah, it'd be different. So that's really the take-home message. If you have something that changes the order of amino acids, it's going to change how those amino acids fit together and how they fold, which is further down the line going to eventually change this and then eventually change this. So you could, by just changing one single amino acid, you could cause this final shape to be completely different. And that will lead to different functions of proteins. Okay, here's something else to think about. It kind of applies this to food. So ceviche. Any of you had ceviche before? Really tasty. If you haven't, you should try it. Ceviche is prepared by marinating fresh raw fish in citrus juice for several hours until the flesh becomes opaque and firm as if cooked. How do you think citrus juice renders the seafood safe to eat? Seems like there's most people either chose A or C. So what is citrus juice? So it's an acid, okay. So if you remember back to last class, we talked about acids and bases. Citrus juice is an acid. Citrus juice is very acidic. What does being an acid mean for citrus juice in terms of chemically? What's the difference between an acid and a base? And something that's neutral. So it's pH, but what is pH a measure of? So this is important to know. Right? We did talk about this last class.
Acidic something?
Okay, yes, circular. It's very circular, yes. Yes, being an acid means that it's very acidic. All right, what does that mean? What is pH a measure of the concentration of? Okay. All right, it's a ratio of hydrogen ions versus hydroxide ions. So something that's acidic has a lot of hydrogen ions. Something that's basic has a lot of hydroxide ions. Hydrogen ions is H+, right? So let's say I take fish, I put it in a lot of H+. A lot of what's in a fish is protein, right? What's going to happen to proteins that are now sitting in a lot of H+? How do you think it's going to change what's going on with that protein? All right, what does that mean? It's going to pull it apart, which is called denaturing, unfolding and inactivating. How's it going to do that? Okay, so hydrogen bond is an attraction between partial charges here. What do you think is going to happen when you throw hydrogen ion in here? Is this amino acid going to be attracted to this partial charge, or is it going to be attracted to this stronger hydrogen? Yeah, it's going to be attracted here, and it's going to break this bond. And same thing here. Same thing would happen if you put it in a strong base, just that our bodies can't really handle strong bases, so it would make us sick. But citrus juice is fine for us. We can handle it. So you are actually breaking hydrogen bonds. You're actually not doing anything to the peptide bonds. Peptide bonds are a covalent bond. They're really strong. They're a lot harder to break. You might break these by cutting the fish, or chewing, something like that. But you're not breaking them when you marinate it in citrus juice. What do you think happens when you cook fish? What does the heat do to proteins in the fish? All right, does the exact same thing. Heat will break hydrogen bonds. So marinating in an acid, cooking it, the exact same end result. You break hydrogen bonds in the proteins, and that's what causes the flesh to look cooked. It's because you are having a similar end product, different way of getting there, but the end result is you're breaking hydrogen bonds. So there aren't ionic bonds that you're breaking. You're not going to break peptide bonds, and you're not dissolving membranes with the citrus juice. You do need to get the citrus juice into it, but it eventually makes its way in. Those ions are pretty small. They'll be able to get into the flesh. So pretty much, more of you chose that than anything else. But
Have you ever eaten that?
oh yeah, it's very good. So it's because
My mom eats it like
Yeah.
all the time. I just don't taste it.
well, you should try it. It's just you marinate it long enough in an acid, it kills everything just like cooking does. So it's exactly the same thing. And it's now just been marinated in this wonderful citrus juice for a very long time, so it tastes really yummy, right? So try it sometime if you get a chance. Shrimp ceviche really tasty, but really any kind of fish, you're just marinating it. It's not tough. Like when you cook things, sometimes the flesh gets a little tough. Here you're marinating in acid. It's tender, flaky. It's really tasty. So. But that's why it works. It's the exact same end result as cooking. So acidic pH denatures, unfolds. Okay. So really kind of a take-home message. It's kind of the next two slides. Protein function is largely dependent on structure, and structure is dependent on hydrogen bonding to make those secondary structures, which lead to tertiary and quaternary structure. So we're going to see a lot of different structures for proteins. In the next few chapters, we're going to talk about structural proteins that make up things like the cytoskeleton. We're also going to talk about contractile proteins, so motor proteins interacting with actin to make your muscles contract. So you can see both of these, myosin and actin, these are both proteins. They have different structures to them. And it allows this one to be a motor protein to actually move, and this one to be more of a structural protein, which this pulls against and allows for muscle contraction. Then we're going to see proteins folded into other shapes, folded into tunnels to allow for transport across a membrane. You can have them folded into these kind of sac-like structures that will allow for storage. You can have them folded into enzymes, so folded into structures that will help allow reactions to go forward at a good rate. So protein's function is dependent on its structure. Structure is dependent on hydrogen bonds. And then when you do things like heat or change the pH to a protein, it's going to mess with its structure. So heat, excessive salt, so ionic changes in ion concentration around it, which is the same as changes in pH in many ways, can break hydrogen bonds, unraveling and inactivating proteins. So taking a protein that looks like this and making it into this. So unraveling it. If you destroy the shape of a protein, you destroy its function. And that's what happens when you're cooking an egg. That's the example here from your book. So you've got proteins in one form, and then when you heat them up, the proteins all unravel and you get a different form. Yeah, well, yeah, that's happening to parts of it. That's why you can kill them by putting salt on them. You know, if you jumped into a vat of salt, it wouldn't be very good for you either. It would, there's other things going on, and we're gonna talk about osmosis later on, and that is also happening. You're changing water balance by changing ion concentration, but I'm sure you're not doing any benefit to their enzymes. Okay, so proteins. That's proteins. Okay. Third kind of thing, different biomolecule that we're looking at are nucleic acids. And I'm only going to really touch briefly on nucleic acids now. We're going to talk a lot more about them later. We're going to really get into DNA and RNA and further a few chapters from now. Nucleic acids are really ideally suited, we know now, to storage and transmission of genetic information. And really, when I talk about genetic information, most of this genetic information relates to how to make proteins. So we just talked about proteins for a long time. Proteins have so many important functions to them. Controlling which protein is found where is really important, and controlling how you make those proteins, because remember, you need to make the proteins in the right way in order for them to fold appropriately and to have the right structure. So nucleic acids will store the information for making proteins. There are two types of nucleic acid. There's deoxyribonucleic acid, or DNA, and there's ribonucleic acid, RNA. And we'll learn there's not all that much different between them, except for one of the sugars that makes deoxyribonucleic acid different than ribonucleic acid. The sugar has one less oxygen, so deoxy, that's what that means. So proteins are polymers of nucleotides. That's the monomer. There's a nucleotide. And a nucleotide consists of three parts. It's got a 5-carbon sugar, which is either ribose in ribonucleic acid or deoxyribose in deoxyribonucleic acid. They have a phosphate group. And they have what's called a nitrogenous base. And there are 5 different nitrogenous bases: adenine, guanine, thymine, cytosine, and uracil. 4 of these are found in DNA. 4 of these are found in RNA. And we'll talk more about that as we get into DNA, really more so later. I don't want you to spend a ton of time focusing on the bases. So really what I want you to know about DNA and RNA, I want you to know the 3 components that make up DNA and RNA. So 5-carbon sugar, phosphate, and nitrogenous base. And then what I'm going to show you briefly is kind of the difference between DNA and RNA as we go into here. So here's what it looks like. Here's what a nucleotide looks like on the top. So 3 components of a nucleotide: phosphate, sugar, nitrogenous base. What will happen, and we'll see on the next slide, is that the sugar and the phosphate are going to form a long chain. The base is going to be on one side. The base is actually what the information is stored in. So there are 5 different bases. Adenine, cytosine, and guanine are found in both DNA and RNA. Thymine is found in only DNA, and RNA only has uracil, but don't worry about that today. We're going to get into this a lot more later in the course. The order of which these bases are found in DNA and RNA is how the information is stored. So we'll actually decode DNA as we go along, and RNA. Nucleic acids form from dehydration reactions. So 2 nucleotides are linked together through dehydration. Here you've got an OH on one end of one nucleic acid, or sorry, one end of one nucleotide. You've got an OH on the other end of the other nucleotide. Dehydration synthesis links them together. So here you've got phosphate, sugar, phosphate, sugar. To break it apart, you've got hydrolysis. If you look, I know this one doesn't have a picture of it. I don't know if I have that. Yep. But if you look at the sugar deoxyribose up here, see there's an OH here and there's an H here. If you looked at the sugar ribose, the only difference between the sugars is instead of H here, you'd have an OH in ribose. So deoxyribose is just missing an oxygen from that one location. But otherwise, they're pretty much exactly the same. So again, I'll leave this until later on this semester when we'll talk a lot about nucleic acids and we'll talk a lot about DNA and RNA and actually look at the process of making a protein from them. But this is enough for now. Just know the three parts. Know that DNA has deoxyribose, RNA has ribose in it. We will see eventually DNA is what's called double-stranded. It's called a double helix. So it'll be two strands, two polymers next to each other with the bases in the middle. The bases are going to form hydrogen bonds between them to link them up. RNA is single-stranded. RNA is a very short-lived polypeptide. It's made and then used, and then it's degraded. Whereas DNA survives for a long period of time. So as long as your cell's alive, the DNA in the cell is alive. But your cells are constantly producing RNA. Right now, as you're sitting here, you're making RNA. And you have RNA that's breaking down right now in every single cell. RNA is like we're going to see, is a copy of the DNA, brings the message to the location where you make a protein. So again, leave most of this for later. Okay. The final biomolecule that I spend a little bit of time on are lipids. Okay. So lipids, I left lipids to last because lipids are a little bit different than the other three biomolecules. So carbohydrates, proteins, and nucleic acids are all monomers and polymers. They all are polymers created by dehydration reactions from single units called monomers. Lipids don't function that way. Lipids are a little different. They are not polymers. Lipids are organic compounds that generally do not dissolve in water. That's one of their main features. And actually in your lab, this is the easiest way to determine if you have lipids present is to put your substance in water and see if it dissolves. If parts of it don't dissolve, you know there's some lipid present in there. So the reason why lipids don't dissolve in water, and we'll look at this in the next slide where I have a picture of a lipid's chemical structure, is that they are dominated by nonpolar carbon-carbon or carbon-hydrogen bonds. So they might have a little bit of oxygen in them. We're going to see something later on called a phospholipid, which has a little bit of an exception to this. But the majority of a lipid is made up of carbon-carbon, carbon-hydrogen bonds. If you remember back to carbohydrates, carbohydrates had lots of OH and lots of OH groups stuck on the outside of it. It was very polar. And we looked at your amino acids. Even if the side chain was non-polar, you still had that amino group, you still had that carboxyl group. Both of those had polarity to them. Amino acids are fairly polar as well. So lipids, non-polar. That's one thing that distinguishes them from the other types of biomolecules. The important lipids that we're going to look at a little bit more today, we're going to look at triglycerides. These are your fats. We're going to look at sterols, waxes, and phospholipids. So we're going to look at a few different kinds of lipids and talk a little bit about their importance within biological organisms. One big thing that I often hear students say, and I kind of cringe every time I hear it, is that they say, oh, lipids are fats and fats are not soluble. You got that kind of backwards. Lipids is a large category for all these nonpolar biomolecules. Fats or triglycerides are simply one type of lipid. So fats are a type of lipid. So are waxes, so are sterols, so are phospholipids. Those are all different types of lipids. So all fats are a lipid, but there's more to lipids than just fats. That's kind of the take home, right? So, all right. So please don't write that anywhere. Oh, well, the reason why lipids are special is that they're fats. No, that's just wrong. Okay, so here's triglyceride. Here are the fats. Here's what a triglyceride looks like. Triglycerides are so named because they have 3 fatty acid tails attached to a glycerol. So they are composed of a single glycerol bound to 3 long hydrocarbon chains called fatty acids. And different fats will have different types of fatty acids. There's different lengths. There's different ways the fatty acids are arranged. But they all have this general structure. And some fats that or triglycerides that you're probably pretty familiar with, butter is made from animal fats, and vegetable oil is made from plant fats, plant triglycerides. There are lots of things you could make butter and oil from, but generally butter is made from an animal fat and vegetable oils from a plant fat. So, if you've ever wondered why is butter solid at room temperature, maybe not our room temperature, sometimes our room temperature is a little bit hot and so it starts to melt, but butter is in general pretty solid, whereas vegetable oil is a liquid, right? The reason why is the shape of their fatty acid tails. So do you notice a difference in their fatty acid tails? Okay, so these are all nice and straight. This one has one fatty acid tail that's bent. And I don't know if you can see it from where you're sitting, but pull up this one, it's blank. Right where it bends here, there is a double bond, and there's another double bond here. So this triglyceride, which makes up butter, has all single bonds between all the carbons. Vegetable oil here has an occasional double bond. And what we refer to those as is a saturated fat or an unsaturated fat. A saturated fat has no double bonds. It's called saturated because the carbon skeleton is saturated with hydrogens. It has as many hydrogens as can fit in there. It makes no double bonds between 2 carbons. Whereas unsaturated fats have 1 or more double bond in their fatty acid tail. Because of this, the fatty acid tails bend, and when they bend, they push 2 neighboring triglycerides away from each other. And now that they're pushed away from each other and have a little gap, they can now move relative to each other fairly easily. So unsaturated fats tend to be able to move relative to each other. Unsaturated fats are well, yeah. This is actually something that if you're taking a nutrition class, you should discuss this with your nutrition professor because there's always conflicting reports and conflicting studies on these about the differences between saturated and unsaturated fats in your diet. Until recently when my wife started telling me otherwise and saying, well, now they think that there's no real difference. My thought was that because these are fluid and because there are double bonds making the fatty acid tails bend, they're easier for you to break down. They also, because they tend to not pack together so tightly, they don't lead to buildup of fats in certain places and lead to problems with that. But probably the most important reason is it's easier for your enzymes to get in here and break them down. Right? Have you heard of trans fats? Okay, so what a trans fat is an unsaturated fat whose fatty acid tails are straight. So again, if you look at this, you've got these double bonds between carbons and it causes a bend to the tail. Here you've got double bonds, but they're still straight. The reason for that is that if you have carbon-carbon, double bond, if you have hydrogens here, carbon-carbon like that, So if you got the hydrogens on the same side of the double bond, this is called a cis configuration versus kind of cram this in here. This is trans, where you have the hydrogens on opposite sides of the double bond. And what happens in a cis is that you now have created this bending of the main carbon chain, whereas with trans, it's alternating back and forth, and it's going to look fairly straight. So trans fat straightens out the fatty acid tails. So at room temperature, is a trans fat going to be liquid or solid? Yeah, it'll be solid. It'll act like a saturated fat. This is why bakers love trans fats, right? Is that they act like you're using butter, so their properties in baked goods are the same as using butter. So you can say you're using these unsaturated fats, but your thing tastes like you're using a saturated fat, and it acts like it chemistry-wise. So cooking is very chemistry-like. But what we found is that health-wise, they act like saturated fats. Sometimes they even act even worse than saturated fats, because they're packed together now. And my thought is that now you have these double bonds, which are really hard to break down. So not only is it harder to access the bonds to break them apart, it's also once your enzymes get there, they've now got to deal with a double bond. So they're really hard to break down. So cis fats are thought to be the most healthy. Saturated fats, pretty unhealthy. Trans unsaturated fats, pretty unhealthy. So that's why they're labeled all the time, right? Yes, it tells you the percentage of saturated fats, but it also tells you percentage of trans fats. Yep.
Haven't most restaurants though ban the trans fat?
Yeah.
Except McDonald's.
Yeah, it's really they're very. There's been enough knowledge about the harm of trans fats that they've been phased out by most manufacturers. I think most people. Yeah. Well, they have to label them now, so you can at least look and you can see. But I think they're probably going back to saturated fats rather than anything else. So, we'll see. Okay, so let me see. I've got. Oh, let's just go. Let's do this one. Okay, here we go. Okay, so let's say you want to make a fat, a triglyceride, solid, more solid. So you want to make sure that it's as solid as you can make it. Which of these would make that fat more solid? So the reason why I asked this question and why I'm kind of focusing a little bit on trans fats and fatty acid tails is that we're going to eventually talk about a type of fat that's very important for cellular membranes, and understanding how you can change the fluidity of fats is going to play a role in membrane stability later on when we start talking about cell membranes. So we'll come back to this in another chapter. Might even just be the next chapter. Okay, anyone need more time to read these? Last few responses. We're over 50% right, so you're doing well. And a lot of people chose the one that was confusing and probably threw you off the most, so that's okay. So let's talk about each of these. Okay, so cis double bonds, does that make something liquid or solid? So here's a cis double bond. What does a cis double bond do to the fatty acid tail? It bends it. And by bending the fatty acid tail, it pushes the fat molecules apart, making it more liquid. So one would be the opposite. That would make something. How about removing hydrogens from a fatty acid tail? So what do you think removing hydrogens does? Yeah, removing hydrogens. So if you have carbon, if I was to remove hydrogens, I'm going to make more double bonds. So that's definitely not going to make it more solid. It might make it more liquid, right? Because you're going to add bends to it. Adding water to the fatty acids. wouldn't really do anything. Maybe if you're talking about a hydrolysis reaction, you could break down the fatty acid. So that's going to, again, make it more liquid. Adding cis double bonds and trans double bonds, they kind of cancel each other out, right? Because the cis double bond is going to make it more liquid. Trans will make it, if you're converting, it's going to make it more solid, or it might just keep the same solidity. So the last one, adding hydrogens and trans bonds to the fatty acid. What does adding hydrogens do? It's going to break up some of these double bonds and straighten out the fatty acid tail. And a trans double bond, again, is a straighter version of this. So if you chose D, you got the right one, which half of you did. Some of you were thrown off by my cis and trans bonds. Because trans bonds, you're right, could make it a little, if you're converting a cis bond to a trans bond, it would make it more solid, but a cis bond would make it more liquid. So they that one kind of cancels each other out. The rest of these are really the opposite. They're going to make it more liquid, make it more fluid. Okay. So just a few more slides for today. I just want to show you a few other kinds of lipids, but really you've gotten the main concept, which is lipids are nonpolar because they have these long hydrocarbon tails. Sterols, we'll talk a little bit about these later on, but sterols are a type of lipid. They're lipids with 4 interconnected carbon rings. And things like vitamin D, cortisone, cholesterol, steroid hormones. There are a lot of hormones that are sterols, and they look like this. They're just carbon rings. Again, primarily carbon and hydrogen. And I talked briefly about this last class, that things like testosterone and estrogen and even cholesterol, they're all very similar. Their only difference are the functional groups that are attached to them. So sterols are fats with this kind of ring shape to them. Other things that your book mentions about fats, lipids, that are pretty important. Waxes are another type of lipid. Waxes are fatty acids combined with alcohols or other hydrocarbons, and they create this stiff water-repellent material. So the waxy compartments of honeycomb or beeswax are this, and they have lots of roles. So waxes being that they are almost water-repellent, and that they have some stiffness to them. They can play a lot of roles in making structures, in making these kind of watertight locations. So a lot of organisms will excrete waxes, and they can then use them. We've used waxes all the time for things, so we use wax throughout our daily lives. Wax is a type of lipid. But really, kind of in the last minute of class, thing I want to introduce, because this will become very important in the next chapter, is a phospholipid. So a phospholipid, the basic structure is fairly similar to a triglyceride, but it only has 2 fatty acid tails. Instead of the 3rd fatty acid tail, it has something called a phosphate group. And phosphate groups are polar. So you've got a polar end, and you've got fatty acid tails, which are nonpolar. So if I was to put this into water, which side, the phosphate side or the fatty acid side, which side would dissolve in the water? The phosphate side, and the fatty acid side would not. So terms that you'll see used for this are hydrophilic and hydrophobic. Hydrophobic would be the nonpolar side, and hydrophilic would be the polar side. So you've got a polar and a nonpolar side. This is going to play a huge role in making a membrane, as we're going to have what's called a phospholipid bilayer. Okay, so there is a nice review in your textbook, and you should check this out at some point. And there's kind of a neat thing on artificial sweeteners. Any questions?